Welcome back. In this video, we're going to continue with section 4.2, the Proteobacteria, starting with the first class, Alphaproteobacteria. Alphaproteobacteria often live inside host cells. In fact, some of these are obligate intracellular bacteria. They need a host because they can't make certain nutrients or energy sources. Some are facultative, which means they don't necessarily need a host, but they benefit further when they do have a host. Some of these alphaproteobacteria are oligotrophs, which means they can survive in low nutrient environments such as the deep soil or even in ocean sediments. An example of alphaproteobacteria include Rickettsia species, and SPP denotes or indicates that there are multiple species within the genus, here the genus being Rickettsia. So Rickettsia are obligate intracellular pathogens, and they need a host or to live inside of a host because they cannot make their own ATP. So the host cell makes ATP and they use it for their own reactions. Examples of Rickettsia species include Rickettsia rickettsii or rickettsii. This causes Rocky Mountain spotted fever, which is a tick-borne disease. We also have Rickettsia Prowazekii, or you can also say Prowazekii, which causes epidemic typhus, which is transmitted by lice. So this infects human endothelium cells, which can cause inflammation of the inner lining of our blood vessels, cause a high fever, abdominal pain, or sometimes even delirium. And epidemic typhus is a severe infectious disease that's common during warfare and when there are mass migrations of people. Alphaproteobacteria also include Rhizobium, which we talked about earlier. These are nitrogen-fixing bacteria that can live in these plant roots, these legume roots. And we'll look at additional genera of Alphaproteobacteria as well. Rickettsia usually requires special staining methods because they're too small to identify with the normal gram stain. And here we can see Rickettsia rickettsii or rickettsii, which causes that Rocky Mountain spotted fever, and they're being shown inside of a host cell. So here's the host cell, the tick hemolymph cells inside the ticks, and you can see in dark pink or reddish, we have our Rickettsia. Table 4.2 provided to us by the book shows some major important genera of Alphaproteobacteria. There are many others that are not included here. I would like you to know these, but especially the bolded ones. Those are the ones we most commonly see in our other readings and other classes, or that show up in the news. Agrobacterium, the first genus here. This is a plant pathogen and can often cause tumors in plants. Bartonella is also fairly common. This is a facultative intracellular bacterium transmitted by lice and fleas, which can cause fever and also, more famously, cat scratch disease. This was a disease that kids used to get a lot. They would get a fever and rash after getting scratched by a cat, and Bartonella does not grow well in the lab, but they identified it eventually by doing DNA sequencing. We have Brucella. Brucella is also a facultative intracellular bacterium transmitted by contaminated milk from infected cows. So it causes brucellosis in cattle and humans, and we can get it from unpasteurized milk, like raw milk. We have down here Rhizobium. We've shown this to you before. These are those nitrogen-fixing bacteria that live in soil. And we have Rickettsia, which we just talked about as well. This is an obligate intracellular bacterium transmitted by ticks, can cause Rocky Mountain spotted fever and typhus. And I wanted to show you an example of those tumors that can be caused by Agrobacterium infecting plants. We often see them when we go walking or hiking. So next time you're hiking or walking around trees, see if you can see any of these tumors. Our first checkpoint. So a common characteristic of human pathogenic Alphaproteobacteria is that they are obligate intracellular bacteria. So they must live and reproduce inside host cells because they cannot produce their own ATP and they depend on the host for energy and nutrients. Our second class are Betaproteobacteria, and these are diverse in their metabolism as well as their environments where we find them. The first genus here is Neisseria. So Neisseria are a type of Betaproteobacteria. Neisseria produce coffee bean-shaped cocci. So they're spherical cells, usually arranged in pairs like this. So often people say they look kind of like coffee beans. Neisseria can cause gonorrhea. So Neisseria gonorrhoeae causes gonorrhea. Neisseria meningititis causes meningitis. Neisseria are microaerophiles. That means they need low oxygen levels to reproduce, to survive. So lower than atmospheric oxygen levels. Bordetella is also a type of Betaproteobacteria. Specifically, Bordetella pertussis causes whooping cough. And these bacteria produce toxins that actually destroy the cilia we have in our respiratory tract. This leads to a severe cough. Burkholderia is also a type of Betaproteobacterium. This is an opportunistic bacterium that primarily infects cystic fibrosis patients, and it can also be presented as a nosocomial or hospital-acquired infection. We also have Leptothrix. This is a type of Betaproteobacterium that is aquatic and can oxidize iron or manganese. This can also clog our water pipes. Now our book also gives us a Betaproteobacteria table, table 4.3, and I'd like you to know these, but especially the bolded ones. Bordetella we talked about. These are the bacteria that cause pertussis or whooping cough. These are aerobic and very fastidious, which means that they're very hard to grow in the lab because they have really specific nutritional requirements. They only grow when we add all of those specific nutrients in the medium, the petri dish or the broth. Burkholderia. So Burkholderia is another one we talked about. This affects primarily patients with cystic fibrosis, or it can be a hospital-acquired infection. And Neisseria we also talked about already. These are microaerophiles that require moisture and high concentration of CO2, and they can cause gonorrhea and meningitis. Here's a picture of Neisseria growing on a petri dish. So be careful, these are not individual cells. These are actually millions of cells that you can see because they're clustered together, growing as a colony on a petri dish. And this agar is called chocolate agar. It is called chocolate agar because of the characteristic color. It's made by heating something we're going to work with in the future called blood agar, which will break down with heat, and the broken down red blood cells will release a bunch of growth factors that these bacteria need to grow. So these are picky bacteria, these Neisseria. They require very specific nutrients to grow well. Here's another checkpoint. Neisseria are classified as microaerophilic because they require low levels of oxygen for growth. They cannot grow well in normal atmospheric oxygen concentrations. So instead they need low or reduced oxygen levels, plus it's better if they have higher carbon dioxide levels. And one type of medium that we use to grow these Neisseria is that chocolate agar I just showed you. So chocolate agar was the agar that is supplemented by those partially broken down or hemolyzed red blood cells. Our next class is Gammaproteobacteria, and this is the most diverse group of proteobacteria. Many are important human pathogens, but others have pretty interesting environmental roles. One major example is Pseudomonas aeruginosa. This is a strictly aerobic, non-fermenting, highly motile bacterium that also forms biofilms. It's also naturally antibiotic resistant, which can make infections pretty hard to treat. It infects wounds, burns, the lungs of cystic fibrosis patients, which is CF here, and can be the cause of urinary tract infections or UTIs. Another member, Pseudomonas fluorescens, produces fluorescent pigments. It literally glows, and we're going to be working with both of these in the lab. For Pseudomonas fluorescens, you'll see it kind of looks like a neon yellowish green color. For Haemophilus influenzae or influenzae, so whenever you see this suffix, you can pronounce it with a hard E or I sound. This one causes respiratory infections, and before we had the vaccine for this bacterium, it was a major cause of meningitis in children. And even though it sounds like flu, it does not cause the flu or influenza because that is actually a viral disease. And here we're talking about bacteria. Haemophilus influenzae can cause both upper and lower respiratory tract infections, including things like sinusitis or bronchitis or even ear infections or pneumonia. Another important genus here is Vibrio. We have Vibrio cholerae, which thrives in basic or alkaline environments like lagoons or seaports. This bacterium makes a toxin that causes severe watery diarrhea, also known as cholera. So it kind of sounds like the specific epithet name here. And the reason for that, for diarrhea, is because of a toxin that this bacterium produces that causes hypersecretion of electrolytes and water into the large intestine. So that leads to really profuse, like a ton of watery diarrhea and major dehydration. We also have Vibrio vulnificus, a different species of Vibrio that causes life-threatening skin infections and bloodstream infections. And finally, we have Alivibrio fischeri, which is famous for bioluminescence. It forms a mutualistic relationship with the Hawaiian bobtail squid. This squid will provide the bacterium with nutrients, and the bacteria help it camouflage through light production. Unfortunately, Vibrio vulnificus shows up in the news fairly frequently. It can cause septic shock, so you can get a blood infection if you get a new tattoo and go into the ocean or beach. It's also fairly common to see it in the news when people get sick from Vibrio vulnificus through eating raw oysters. So raw oysters can be contaminated with Vibrio. And here's Alivibrio fischeri, that bioluminescent bacterium that lives on the squid and helps protect the squid from predators through camouflage. Legionella pneumophila is another example of Gammaproteobacteria. It grows really well in warm water, unfortunately in systems like air conditioners. And when it's aerosolized, it can spread and cause Legionnaire's disease, which is a type of pneumonia. In fact, these bacteria got their name, Legionella, from the first known outbreak of Legionnaire's disease. And this happened in a hotel that happened to be hosting an American Legion Veterans Association meeting in the 1970s. Next are our family Enterobacteriaceae, which are enteric, which means intestinal bacteria. And these are facultative anaerobes that ferment carbohydrates. I haven't really talked about these vocab terms yet. They are from your prereqs to this class, but we're going to learn more about them soon. I have them in my slides so that when we start the Metabolic Unknown project in lab, you have them as reference. You have this information. For the Enterobacteriaceae family, there are two subgroups, coliforms and non-coliforms. Coliforms are named after the most common one, E. coli or Escherichia coli. These coliforms ferment lactose completely. Some of them are mutualistic and benefit us, but some of them are disease-causing, such as the strain E. coli 0157H7 that produces a toxin known as Shiga toxin or Shiga toxin. So Shiga toxin interacts with our ribosomes and stops protein synthesis. When we can't make proteins anymore, that leads to cell death and hemorrhagic colitis. And you'll see that is characterized by inflammation of the GI tract and also bloody diarrhea, and it can be fatal. For non-coliforms, these don't ferment lactose at all or only incompletely ferment lactose. And examples in this category include Salmonella, which causes foodborne illness or food poisoning. Shigella causes dysentery, and you'll also see diarrhea there. And Yersinia pestis, which causes the plague and is often carried by fleas found on rodents like squirrels. And here's the table from our book showing the different genera from the class Gammaproteobacteria. You want to know the previous ones in the slides that I just talked about, as well as the bolded ones. So Enterobacter, we're going to see these in our lab soon. They can cause respiratory tract infections and also UTIs. We have Escherichia, which I just talked about, such as E. coli. We talked about Haemophilus and Legionella already. Klebsiella we're also going to be working with in the lab. We're going to see that this can cause, or these bacteria can cause pneumonia. Proteus. For Proteus, we're also going to be working with these in the lab. They are common in our GI tract, but can also cause UTIs and even blood infections. We also talked about Pseudomonas, Shigella, Vibrio, and Yersinia. We also want to be familiar with Serratia. We'll be working with these in the lab. You might see that they produce a red pigment at certain temperatures, and these can be responsible for many different types of hospital-acquired infections. For our Gammaproteobacteria checkpoint, we talked about many different types of bacteria, and some of them were in particular families. So one family that's important are the Enterobacteriaceae. So remember, these included coliforms and non-coliforms. Coliforms included E. coli or Escherichia coli. Non-coliforms were bacteria like Salmonella, Shigella, or Yersinia. For another family, there's a second family that we saw some bacteria within, and this family, Pseudomonasaceae, includes bacteria like Pseudomonas aeruginosa. So these were those antibiotic-resistant bacteria that can form biofilms. Our next class are the Deltaproteobacteria. These are less common but still really interesting. These include sulfate-reducing bacteria, abbreviated SRBs, that use sulfate in their electron transport chain as the final electron acceptor instead of oxygen. So sample bacteria from this class include Disulfovibrio orale, which is linked to gum disease, also known as periodontal disease. So something kind of like that inflammation there. We have Delovibrio, which I don't have a picture of, but this one's interesting because it attacks other gram-negative bacteria. And then finally, we have Myxobacteria shown on the bottom right here. These are found in the soil, and they're kind of similar to how mushrooms work in that they form multicellular fruiting bodies. So we know that bacteria are single-celled, right? They're unicellular organisms, but in some, like Myxobacteria, they can come together, grow together as a fruiting body, and also produce dormant myxospores. And here's the table from our book with Disulfovibrio orale, probably the most clinically relevant. As we mentioned, they can be linked to gum disease. For our Deltaproteobacteria checkpoint, remember Myxobacteria are the type of Deltaproteobacteria that form fruiting bodies. These soil-dwelling bacteria, they're motile, and they're actually very highly social. So they can aggregate, they can come together to form those multicellular fruiting bodies, and they can also produce those dormant myxospores. Those get spread into the air, land in different places, where they can eventually form more fruiting bodies. Epsilonproteobacteria are our smallest gram-negative class, and these are microaerophiles. They need low oxygen levels to survive. And there are two examples of important human pathogens here. Campylobacter jejuni is a foodborne pathogen. We often get it from undercooked poultry, like undercooked chicken, and it causes severe enteritis, which is inflammation of the small intestine. Another one here is also Helicobacter pylori, shown here. This is a bacterium that can survive the acidity of the stomach, and they do so by producing an enzyme called urease that can help make the stomach a little bit less acidic. Helicobacter pylori is the most common cause of chronic gastritis and peptic ulcers, and it's also linked to stomach cancer. All right, and in the table from our book, you can see Campylobacter and Helicobacter. These are genus names that are part of the class Epsilonproteobacteria. So Barry Marshall and Robin Warren are the scientists that won the Nobel Prize in 2005 for their discovery that the bacterium Helicobacter pylori causes ulcers. And an interesting or kind of fun fact is that they were doing their work in the 1980s where they found that I think they studied around 13 patients with duodenal ulcers, And they found that all of those patients, 100% of those patients, were infected by Helicobacter pylori. So their hypothesis was that this bacterium causes the ulcers. And at the time, people didn't really accept that. They thought Helicobacter pylori were harmless commensal bacteria. And so in response, Barry Marshall here on the left, he actually drank a petri dish culture full of Helicobacter pylori to prove that the bacterium can infect a healthy person and cause gastritis. So don't do this at home, right? Don't drink or eat your bacteria that you grow in the lab, but in this case, it worked out for them. For our Epsilonproteobacteria checkpoint, remember we learned about Campylobacter jejuni that causes foodborne illness, and we can get it from undercooked poultry. It causes severe enteritis, which was inflammation of the small intestine. And the second one we learned about were Helicobacter pylori that survives stomach acid, can cause chronic gastritis, peptic ulcers, and can be linked to stomach cancer. So I'll put these two together and discuss them at the same time while we recap 4.2. We had our first class, Alphaproteobacteria. These were obligate or facultative intracellular bacteria. Some of them were oligotrophs that could survive in low nutrient environments. An example could include Rickettsia rickettsii, which causes Rocky Mountain spotted fever. We also had Betaproteobacteria. These had different types of metabolic strategies, and they could live in different types of environments, including inside us as pathogens. So one example is Neisseria gonorrhoeae that causes gonorrhea. The next class, Gammaproteobacteria. So these were the largest and most diverse class. These included many pathogens, like our enteric bacteria. Some of them formed biofilms. Some of them were bioluminescent, like we saw in that squid. So one example could be Escherichia coli, right? E. coli. That's has a mutualistic relationship with us in our gut, but we had some strains that were pathogenic. We have Deltaproteobacteria. These included the sulfate-reducing bacteria. Some of them were bacterial predators. Others could live in the soil and form fruiting bodies, like the Myxobacteria. And then our smallest class are Epsilonproteobacteria. These are microaerophiles. They require low oxygen levels to grow, and some of them were pathogens in the GI tract, like the one we just talked about, Helicobacter pylori. That takes us to the end of our second video. In our third video, we'll talk about non-proteobacteria.